Hello friends, welcome to my channel Clinical Biochemistry by Dr. P.K. Prabhakar. Today I am going to talk about karyotype and karyotyping. So what we will do, slowly first we will understand about what is karyotype and what is karyotyping, and then we will talk about how this karyotyping is going on, what is the detailed process. So there's different steps are there. So we will talk about those steps and how this karyotyping, what is their significance, how it is going to happen. So all those things we will talk one by one. Okay. Uh, so let's start. So as you know that what is karyotype? So probably you might have heard about karyotyping. Karyotype. The karyotype is a process of preparing, arrangement, categorization chromosomes using cell culture techniques. So we are doing cell culture, and then we are going to do this karyotyping process. So that is this. So there we are going to do four steps, four things, like we prepare the cells so that chromosomes will be analyzed, chromosomes will be evaluated, chromosomes will be arranged, and then we are going to arrange them according to the size and categorize it. So that is called as karyotype, and the process of this, all these steps are called as karyotyping. Okay. So let's go to the PPT first. Good evening, everyone. Okay. So if you'll see the karyotype, let's introduce about the karyotype. Uh, the karyotype method is entirely different from the PCR. So whenever we are talking about the karyotype, it's related to the DNA, but it is not the PCR. It is different from the PCR, different from the DNA sequencing. In the case of karyotyping, instead of gene sequencing, we are doing studying the entire chromosomes. Whenever we are talking about DNA sequencing, we are using a part of DNA. In the PCR also, we are taking a fragment of DNA. But in this karyotype, we are going to talk about, we are going to say whole chromosome, complete chromosome. So here, using a cell culture methods or a standard karyotype or karyogram, a person or organism can be prepared. Okay. Now, so the karyotype of is an individual's collection of complete chromosomes. All the, like if I'll talk about human, so in our case we are having 46 chromosome. So this is the collection of all the 46 chromosome. This karyotype is used for the looking for abnormal number of chromosomes or the structure of chromosomes. Means there are three different things we are going to see here. Either chromosome number has been increased or it is going to decrease. Like that, we are going to call it aneuploidy, where chromosome number will be increased not by complete set, by numbers. Either 46 become 47, 48, or 46 become 44 or 45, like that one. That is abnormal number. So there we are having nullisomic, monosomic, double monosomic, then trisomic, double trisomic, tetrasomic, and all those things. Or there is a insertion or deletion of some fragment, some portion of the chromosomes. That is the change in the structure of chromosomes. So karyotype give us information about the abnormal number or abnormal structure of chromosome. This test can help us to identify genetic problem which causes a specific disorder or a disease. Karyotyping is a test for examining chromosome in a sample of cells, and this term is referred to the laboratory techniques that produce an image of an individual. Okay, so to understand the process, specific to precise what we have, first understand the chromosomes. Each pair of chromosomes have a different means every chromosomes in our case, in our cells, are different. They are differ in their size, their shapes, their genetic materials. So here we are having 46 chromosome, not rather we are having 48 chromosomes. So these are from here to here, it is 22 pair, and then we were having 22 pair, then we are having XX or XY depending on male and female. So each chromosome is going to differ in their size, shape, and genetic material, genetic constitutions. Some chromosomes are larger in the size and have a huge amount of gene and the genetic material, while some are having the fewer, smaller size, smaller genes. Like if you see this 46 chromosome, the chromosome number 1 is the largest in the size. That is the largest. So this is according to the size arrangement. This is one karyotype we are having. So here, based on the amount of genetic material or number of genes, the size of each chromosome is different. Like I have told you, chromosome number 1 is the largest and contains the maximum number of genes than any other chromosome. Okay. Several factors are there. That factor can be intrinsic factor or extrinsic factors. Changes the composition or structure of chromosome. Those are either replication errors, non-disjunctions, different kind of radiations like UV rays. Using conventional cytogenetics techniques like karyotyping, we can study chromosome of any organism. Okay. Now come to the karyotyping. If you go the term karyotype, the word karyotype is coming from the Greek word. The word is derived originally from the karyon. The word is coming from karyon. Karyon, which means nucleus or the seed. It is no way associated with either DNA or genetics. The reason why this karyon has been chosen for this type of study is the chromosome were first observed in the plant nucleus. So that's why this term has been given to the karyotyping. Okay. So the chromosome were first observed in the plant nucleus. That's why this karyon word has been given for this study. The word chromosome was also derived from the Greek word chromos, which means color, and the soma means body. So there we are having chromosome. Okay. Now this here we are having a picture which shows us this is the different cell division. So normally we are having two types of cell division, as everyone is knowing, mitosis and meiosis. So in this case, this is the interphase, prophase, metaphase, anaphase, and telophase. And then after telophase, cell is going to divide. Again, it is going to enter into resting phase or then interphase. The chromosome number is going to duplicate in the case of interphase, and the maximum visible, most condensed structure of DNA is in the metaphase. So that's why we are going to use metaphasic DNA for our study. Here chromosome will be visible in the most prominent. If you see each chromosome, their chromosome, this is one chromosome, this is which we are going to see here. This is one chromosome which is having two chromatids. This is two sister chromatids. Later on we will see. So this is one chromosome which is having two chromatids. This is one chromatid, this is second chromatids, and both chromatids are almost equal. They are coming from the different places, different parents, and their content will be exactly same. How we are going to count the chromosome numbers in our case? So if you see here, so this is a normal cells where we are having, this is the chromosome number as we are going to see in this form. So this is case we are having 46 chromosome. So in the interphase, when chromosome is going to be duplicated and both duplicated portions is joined at the centromere places. So in that case, chromosomes looks like this one till up to metaphase. So the same chromosomes which is duplicated in the duplicated forms, in the double form replicated forms associated at the centromere positions, and it will be up to metaphase, and that will be, we are having 46 chromosome. After metaphase, in the anaphase, when both the chromatids will be separated from here. So one chromatid goes this side, another chromatid comes this side. So in that case, we are having 92 chromosomes in the anaphase. It means you can see visually 92 chromosomes in the anaphase if you want to see it. After, so up to metaphase, we are having two chromatids which will visualize. In the case of anaphase, we are having again a single chromatid because it has been separated. So when we are going to see the chromosomes and chromatids, this is what we are going to see of the metaphasic chromosomes only, because at that place, that portion, it will be most visible form, most condensed form. So normally metaphasic chromosomes is only going to be visualized. Okay. Now in the chromosomes we are having one short arm, one and the long arm. This is not for every cases. Some chromosomes where a centromere is exactly at the center. In that case, both portions, both the hands, both side will be equal. But in some cases, most of the cases we are having unequal distribution. So one side of the centromere we are going to have a short arm, another side we are having long arm. So the short arms normally we are going to call P arm, and the short long arm we are going to call it Q arm. And both side we are having telomere here. Okay, so P arm shorter arm, and the Q arm is the longer arm we are having. If you'll see this chromosomes, whenever we are going to study any chromosomes, so that time we are having this type of structure, this type of things we are going to talk about. Means chromosomes have different chromatids, and the chromatids have, means those chromatids we have one longer arm, one shorter arm. Short arms we are going to call P arm, and long arm we are going to call Q arm. These short arms or chromatids are going to be divided into regions. Then regions will be divided into bands, which is also called as sub-regions, sub-bands, and sub-sub-bands. So if this is the chromosomes or a gene's positions, locations, this can considered as this will be 6 is the chromosome number. P means I'm talking about the short arm. Then 2 is the region number 2, sub-region or band 1, sub-band 3, and sub-sub-band 4. For example, if we'll say this chromosome, CFTR gene, which normally we are going to have a mutated CFTR gene in case of cystic fibrosis. That CFTR gene's location is 7q3131.2. What does this mean? This means this CFTR gene is present on chromosome number 7. Then it will be on the long arm Q. Then it will be present on the region number 3. In the region number 3, it will be band 1 and sub band 2. So this is how chromosome locations is going to be visualized. How this band, sub bands, regions is going to be visualized. This is on the basis of staining, on the basis of coloring. So we are having different banding processes. Like we maximum, most commonly, most frequently, we are going to talk about G banding, Giemsa staining. So there we are going to GTG banding, that we will talk later on. So on the basis of those banding patterns, we are going to see the different regions and different location of the chromosomes. So how scientists identify different chromosomes? So there are 3 different criteria, you can call it. Those criteria are the size of the chromosome, which can be visualized, and then we are going to have centromere positions, and then we are having banding pattern, staining pattern. The size is the easiest way to differentiate 2 different chromosomes, because you can just arrange it. We are having karyogram, and then from there you can see the size, and you can differentiate 2 different chromosomes on the basis of their size. Then on the basis of centromere positions, also chromosomes can be divided, can be grouped. And then we are having banding patterns where Giemsa band or other different kind of fluorescence dyes you can use it. And then we are going to differentiate it. Is it clear? If anyone is having any question, you can ask. Okay, fine. Now, size, according to size, whenever we are going to do a staining, we are going to see the size, and then we are going to identify it. When we are talking about the chromosome centromere positions, so normally we are having 4 different types of chromosomes. Yeah, chromosomes can be grouped into 4 different categories depending on the centromere position, centromere places. So if centromere is placed at the center and both sides of the centromere we have equal hand size will be same, it is called as metacentric, means centromere will be in the center. If centromere is not placed in the center, it will be one side, slightly one side, so that one we will have one large hand, one short hand. Then we are going to call submetacentric. Acrocentric is when we are having this unequalness is too much, means one arm is very short, another arm is very long. That will be acrocentric. And when this centromere is at the terminal place, so that short arm is almost nil, no short arms will be there, that is called as telocentric. In some cases you can find subtelocentric also, like we are having submetacentric, similarly we are having subtelocentric. So total 5 different category we can have. Now if you see, all the human chromosomes, 46 chromosomes, are going to fall into 3 category only. They are either metacentric or submetacentric or acrocentric. Telocentric and subtelocentric, we don't have any chromosomes. So chromosome number 1, chromosome number 3, chromosome number 16, chromosome number 19 and 20 are metacentric, means their hands, both P arms and the Q arms, will be equal in the length. Then we are having maximum chromosomes falls into the submetacentrics, where chromosome number 2, 4, 5, 6, 7, 8, 9, 10, 11, 12. So up from 4 to 12 continuous. Then chromosome number 2, 17, 18 and X chromosome are submetacentric. And chromosome number 13, 14, 15, 21, 22 and Y chromosome are acrocentric. And we don't have both telocentric and subtelocentric chromosomes have not been observed in case of humans. So we are having our chromosomes are going to fall only in these 3 categories. Okay, there is another classification depending on the chromosome number, centromere places and their locations. So the size of the chromosomes, we are having chromosome number 1, which is large chromosomes and metacentric, that is chromosome 1 to 3. Then group B, which is a large chromosomes depending on size and some metacentric, that is 4, 5. Chromosome number C, that is medium in the length, means slightly smaller but some metacentric, 6, 7, 8, 9, 10, 11, 12 and X. Group D, medium, acrocentric, 13, 14, 15. So this is another type of classification. So both type of classification you can see depending on the centromere locations and centromere positions. Then come to the banding pattern. How banding pattern is going to happen and how we are going to use it. So the banding patterns are the pattern of light and dark transverse band on the chromosome. There we are having alternative dark and light band. The light and dark band becomes apparent by staining the chromosomes with the chemical solutions, then view under a microscope. So different depending on the which type of dye, which type of stains you are going to use, similar type of microscope you can use it. So chromosomes are stained with very different kind of dye. I have told you some kind of chemical dye, some kind of fluorescence dye we are going to use, and that enables the chromosome segment to be identified. These bands describes the location of genes on the chromosomes. Just now we have seen CFTR gene on the chromosome. Most of the dyeing method, banding patterns can visualize more than 550 bands in the per haploid set. But if you are going to use more high resolution methods, you can visualize up to 850 bands per haploid set. These are the different banding patterns we can have. That is C banding pattern, which is also called as constitutive heterochromatin staining methods. G bands, that is Giemsa staining. Q bands, that is quinacrine band. R bands, reverse Giemsa staining, and T bands, telomeric bands. Other than that one, we are also having few other banding patterns. So one by one, few characteristic features we will see. Most commonly, we are going to use Giemsa banding pattern. So first, G banding or Giemsa banding, that is exactly the same, or this is also called as GTG banding. Why GTG banding? Because in this case, first we are going to use Giemsa, we are going to use trypsin, and GIMSA, so it is called as GTG banding. Okay, so this GTG banding or GIMSA banding is useful for identifying genetic disease through the photographic representation of the entire chromosome. Here we are going to use metaphasic chromosomes are treated with trypsin, so that's why GTG banding. This trypsin normally going to, it is a proteolytic enzyme, so it is going to digest the proteins, and chromosomes will be little bit more relaxed so that it will take the stains. Otherwise, chromosomes will be associated with the proteins, so there it will not be taking too much dye. Once you are going to treat with the trypsin and then you are going to stain with the GIMSA, so there we are having two different regions we are going to see it. One region which we are going to call it heterochromatin region. Sorry. One we are going to call it heterochromatin regions, which tends to be rich in adenine and thymine. Okay, and they are relatively gene poor. They are non, you can call it transcriptionally inactive. They are going to take more darkly in the G banding pattern, so they will be more intense colored. Then second portion, second part is that is less condensed, little bit more relaxed. That is called a euchromatin region, and they are more rich in GC, guanine and cytosine. They are transcriptionally active, and they incorporate less GIMSA stain, so they will be visualized in the dark light staining. Means dark staining is heterochromatin, and light staining is in case of euchromatin. The pattern of bands are numbered on each arms of the chromosome from the centromere to the telomere. So we are counting starts from the centromere and move toward the terminal end of the chromosomes. This numbering system allows any band on the chromosome to be identified and described precisely. The opposite of the reverse G banding is called as reverse banding, R banding, where euchromatin takes intense color and heterochromatin will be taking light color. So banding can be usually identified and gives you chromosomal abnormality informations depending on their banding patterns. Then we are having Q banding patterns. Q banding. Here, in place of normal GIMSA staining, we are going to use quinacridine fluorescent dye for the banding techniques. And in this case, it is almost very similar to G banding patterns. Only difference is in case of G banding, we are going to use normal dye, that is GIMSA. In case of Q banding, we are going to use quinacridine fluorescent dye. So we have to visualize under the fluorescent microscope. So this quinacridine binds to the AT-rich heterochromatin regions and emits yellow color fluorescence. Due to this distinct banding pattern, chromosomes are seen under the fluorescent microscope. That is the only difference. So because this quinacridine binds to the AT region, so AT region heterochromatin region will give you yellow color. Then we are having C banding. C banding is also called as centromeric banding, in which only the centromere of the chromosomes is going to be stained. So only centromere will be stained. So it is also called as centromeric banding pattern. The present banding techniques is a specialized modification of the G banding, which only stains the centromere region of the chromosome. However, the C banding methods is not so oftenly utilized for cytogenetics and diagnostic purposes. So this is not very frequently used. And the entire process of getting the C banding is different from the conventional G banding. Then we are having three other banding patterns, that is R reverse banding, NOR banding, and the T banding patterns. R banding, the R banding is called as reverse banding pattern techniques. It is a reverse of the G banding. Here, slides are first treated with the phosphate buffer followed by the G banding to produce a inverse banding pattern. So inverse means here heterochromatin region is going to take the light color and the euchromatin region is going to be darker. That is called as reverse banding. Then we are having NOR banding. NOR banding or the nuclear organizing regions. Here we are going to use a silver staining methods and majorly used to detect the acrocentric chromosomes. And then we are having T banding, that is a less popularly banding methods in the T banding, which is used to study the telomeric region of the chromosome. So these are the different banding patterns. The more commonly we are going to use, more we are going to visualize chromosomes is G banding, that is Giemsa banding. Okay. Now, which type of cells? What will be the sample for this banding? So you can take any nucleated cells because every nucleated cells will have the chromosomes and there that can be used. But most commonly we are going to use lymphocytes. You can also use skin cells. You can use tumor cells. For fetus, we can use amniotic cells, chorionic cells. So these are the different cells used for chromosomal analysis. Now if you see the how we are going to start the process, when we are going to want, if you I want to do the karyotype, so there are few steps. What are the steps for karyotypic age? First, a cells, that is somatic cells, which is the actively dividing cells, is going to be taken, as we have seen the samples. So you can select any sample cells which is actively dividing, but most commonly we are going to take lymphocytes. Then these cells will be treated with a drug to induce mitosis. So cell division will start, and then cells are grown in a culture for many days, several days. After this one, cells are treated with a chemical which is going to stop mitosis in the metaphase. So cell will be arrested in the metaphagic position of the mitosis. Most commonly, we are going to use colchicine to arrest the cell division. One, so cell will not go for anaphase; it will be in the metaphase only. Then these cells are chemically preserved and spread on the microscopic slides. There we are going to stain it and visualize it. After this one, cells are viewed with a microscope equipped with a digital camera, and that is if you are going to use the different kind of thing, depending on the color, we are going to use a microscope. Chromosomes are displayed in a computer monitor, and then a software we are going to use. That software is going to arrange these chromosomes according to the size and their shape. So ultimately, we are going to have a karyotype. So that karyotype is a visual display of a pair of condensed duplicated chromosomes. Because if you see, these are the pairs. So if you see the single chromosomes, this chromosome actually it is a two, means duplicated chromosomes. It is the which is not divided. So two chromatids in each chromosome. So overall, we are having 92 chromosomes in the karyotype, but which is going to be divided after the metaphase in the anaphase. So it will be divided, but in the metaphase we are having two chromatids, that is duplicated chromosomes arranged by the size, shape, and banding pattern. So this is the complete set of metaphagic chromosomes. So this is the overall different steps. So first we have collected the samples. Now if you want to see the detailed study of means detailed on the sample collection, any body cells uh, which is going to divide, actively dividing cells, can be cultured. However, blood cells, amniotic cells, solid tissue, tumors, chorionic villi cells are the most common samples using for karyotyping. The sample is collected in a sterile tube uh, or heparin tube or if F collecting tubes. The sample is immediately sent for a karyotypic center. Transport the sample under the 4-degree temperature. It is essential to maintain the aseptic conditions. Sorry. Sample collection also as crucial as any other techniques in karyotyping. The blood sample is collected in heparin tubes. EDTA prevents cell division and therefore not used. Then cell culture, that is the second step. Cell culture, normally it is a mammalian cell culture. The sample is cultured using the culture media. Different types of culture media we are going to use. Most commonly we are going to use RPMI1640 cell culture media. Cell culture contains number of things. It also contains colchicine and number of other components. Antibiotics, staining, these are the different things we require. Then we are going to prepare the metaphasic chromosomes. In the metaphase, chromosomes are arranged well, most condensed structure. So we need that one for karyotyping. To fulfill the present aim, we need to arrest the cell under our cells in the metaphase stage of the cell division. So most commonly, I have told you, we are going to use colchicine into the culture to prevent the spindle formation, and therefore cell division is going to be arrested at the metaphase. Sorry. Then we are going to use hypotonic treatments. This, in order to isolate chromosomes, we need to swell cell to observe chromosome. So that's why we are going to use hypotonic treatment. So hypotonic treatments, normally their water will enter into the cells and cell will swell up. So a special type of solutions, that is hypotonic, is going to be utilized to swell the cells, so that chromosomes will be separated on the slides. Then we are going to do chromosome harvesting. Now we need to harvest the cells to remove the debris. Using this treatment of methanol and glacial acetic acid with the process of centrifugation, cells are harvested. Then we are having slide preparation, that is the sixth step. In the slide preparation, the other slide preparation methods, the slide preparation techniques in karyotype is slightly different. Here, to separate every chromosomes properly, we need to drop a cell suspension from some height, so that the pre-chilled methanol treated cell slides is used, so that once the cell suspension hit the slide, it is immediately fixed on the surface. After that one, we are going to stain it for banding patterns, staining for visualizations. So there we are having different type of staining. Most commonly we are going to use GIMSA or G staining. Chromosomes are stained with blue for in GIMSA and looks like uh, bluish in the color. Then we are having microscopy, where we are going to visualize it. So the slides are observed normal under the 10x microscope lens to choose the best and well stained field. Then it is observed under the 45x and the 100x oil immersion lens. So roughly 50 to 100 well separated metaphase chromosomes field are observed to validate the result. So this is the whole process, and this is the sequential event. So we are going to collect from there. We will take the WBC or lymphocytes. Uh, there we are going to use phytohematoglutinin in the cell culture. So cell will be cultured. Then in the culture medium, we are going to use colchicines and hypotonic solutions, so that cell will swell up and chromosomes will be visualized. Cell will be fixed with methanol and other process in the slides by dropping. And then we are going to use heat with trypsin and the stain with GIMSA. And then we are going to analyze the cells, and then software will arrange it in the form of karyotype. This is the whole process of karyotyping. When we are going to talk about the fetal karyotyping, in that case, normally amniocentesis. So we are going to take cells from amnion or chorionic cells. So there, through amniocentesis, that is a process, but through injection, we are going to collect the amniotic fluids. From there, we are going to collect the cells. Cell will be cultured, and karyotyping will be done, and result will be analyzed. So this is the whole process of karyotyping. Now, if you'll see the advantages of karyotype, what are the advantages? What are the disadvantages of karyotype? The cytogenetic techniques, especially the karyotyping methods, is utilized to observe chromosomes. In a PCR, we can amplify the entire genome, because in PCR we are going to use only a specific portion of DNA to be amplified. So here in karyotype, we are going to use whole complete chromosomes to be studied. To study the chromosomal aberrations, we have to perform karyotyping. However, nowadays FISH, that is fluorescence in situ hybridizations, special karyotyping, microarray-like techniques are available. But again, karyotyping is having their own significance. The present method is powerful enough to identify numerical and structural chromosomal abnormality. So it gives us either any kind of aneuploidy or any kind of structural translocation or any kind of deletion in the chromosomes. It is a bit accurate and thus used to prenatal diagnosis and screening of chromosomal abnormality. So these are certain advantages. At the same time, it is having certain disadvantages also. The disadvantage of karyotype are more than advantages. It is a time-consuming process. That is the most drawback. High-end sophisticated instrument is needed. Strict aseptic conditions are required throughout the protocol. The chances of contaminations are very high, because we are going to do cell culture also, and there we are having maximum chances of getting contaminations. The chance of reaction failure is very high. A high level of expertise is needed to process and analyze the result, and also the expert touch is required to interpret the result. So these are disadvantages. Three different important information what we can get from the karyotype is the missing chromosomes, like chromosome number has been increased or chromosome number has been decreased. So either the extra chromosomes or the missing chromosomes. Then any portion of chromosomes has been deleted, duplicated, or translocated. So any kind of structural changes in the chromosomes that can also be studied. Now if you see the application of chromosomes, uh, so first, to study chromosome number and structure, the study of telomeric regions, to identify chromosomal abnormality, to study centromere locations, then to identify the relative size of the chromosomes, and to differentiate the chromosome of different species. So these are the six important applications of the karyotyping is there. Now I will show you some of the karyotype for some of the number, when number has been increased, that when chromosome number has been going to be increased by one, that is called as trisomic, mean in place of 46 we are going to have 47 chromosome. So if chromosome number 121 is going to be increased by one, that is called as Down syndrome. If chromosome number 18 has been increased by one, in place of 18 pair we are having three chromosomes there, it is called as Edward syndrome. If chromosome number 13 becomes increased, that is Potter syndrome. Then chromosome number 8, that is Warkeni syndromes. Similarly, it can happen in the sex chromosomes also, like sex chromosome in place of two double X, it becomes triple X, or XY, it becomes XXY, that is Klinefelter syndromes, or XYY. These are the different trisomy of the sex chromosomes. Like here you can say, in this case, this is chromosome number pair 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21 pair. Here normally in 21st pair, we should have only two chromosomes, but here we are having three chromosomes. So this is trisomic for 21st. So 21st trisomy, that is Down syndrome. So this is Down syndrome's karyotype will looks like this one. At the same time, this is Edward syndrome's karyotype, where chromosome number 18 pair will have three chromosomes. Similarly, this is Klinefelter syndrome, where in place of XY we are having XXY. That is, these are the different examples of the karyotyping. So this is all about for karyotype and karyotyping. Now, if you are having any questions, anyone is having, you can ask me, and I will be happy to give your answer. Otherwise, you can follow me, follow my channel on any of these platforms. If later on also, if you have any questions, you can write in the comment box, you can write in the chat box. I will be happy to give your answers. And if you have not subscribed my channel, you can subscribe it. If you like the video, press the like button. You can share with your friends. And thank you very much. Have a nice day. Any question anyone is having? Okay. Thank you very much.